Hello again, students. We are moving right along at chapter 16 today. So this is looking at evolution and how populations evolve. So we're going to look at some Hardy-Weinberg equilibrium, talk about how populations change over time, things like that. So first talking, or just kind of a general outline, genes, population, evolution, natural selection, and maintenance of diversity. So population is a group of organisms that are living in the same area. And then population genetics is the study of the diversity in terms of allele differences. Now, before I got into education, I actually worked at a zoo for 7 years. Maintaining the genetics of a group of animals was very important in the zoo field. We were not allowed to breed certain animals unless they were the genetics were different. So we had what was called a species survival plan, and the genetics of the animals were very closely watched. If, like, for instance, we had lions and giraffes at our facility, and the lions and giraffes were genetically different enough that they were considered a good match. So we had permission to breed them. But there were some animals that were too similar, so we did not breed them, and we either used contraception or we had to keep them in separate enclosures so that they did not interact. So there were different methods that we used to keep them separate. So that goes into population genetics, making sure that the right genetics were utilized and other genetics weren't. So it is something that we pay attention to in zoos, but in nature it's not something that is obviously watched as closely. So it evaluates the diversity of a population by studying the genotype and phenotype frequencies over time. If the population is healthy, it's not something we have to worry about, but if an animal is endangered, then we have limited genetic populations to pull from, such as the black-footed ferret. The black-footed ferret is a really good example of this, and it experiences a bottleneck effect, which we'll talk about later on. So most of the time we experience something called microevolution. There's microevolution and macroevolution. Microevolution is something we experience much more often. Micro is on a smaller scale and does not result in the creation of a new species. It is a change that we are observing within a species, not creating a new species, but a change in like characteristics that is just within a species itself. So think of like dog breeds, for example. We can see dog breeds change. It's still a dog, but we can see those dog breeds change over time. And like golden doodles have become so popular lately, but a golden doodle is still a golden doodle. It's still a dog breed. Can breed a golden doodle with any other dog and get a different type of dog breed. But that's an example of microevolution. Pertains to evolutionary changes within that population. Variation alleles at the gene loci. Remember, loci is a location, and all individuals make up the gene pool of that population. A gene pool of a population can be described in terms of the frequency, how frequent we see that allele come up. And the allele frequencies is just how often we see a certain trait appear. So homozygous, heterozygous, and homozygous recessive and homozygous dominant. So this is just basically how often we are seeing these traits appear within the population. That's what the frequency means. So how often are we seeing the homozygous recessive appear, and how often are we seeing the homozygous dominant appear? That's what that means as far as the frequency. And these are the loci, of course, the location on the chromosome itself. So allele frequency is the proportion of each allele within a population's gene pool. So a gene pool is we're looking at overall genetics that species carries in that population. And it's sometimes hard to determine and maybe impossible to determine, but we can guess and we can use math to help us estimate what the frequency is within a population. And this relationship is described by P plus Q equals 1. P is the frequency of one allele and Q is the frequency of the other allele. 1 is equal to, think of 1 as like 100 or 100%. So if you add 50 plus 50 equals 100%, or 20 plus 80 equals 100%. That might be another way that you can kind of think of it. So microevolution involves a change in these allele frequencies within a population over time. So again, it's changing the frequency of these alleles. It's not making a new species. It's just changing, like, for instance, how many gray owls versus how many brown owls you're seeing in this population over time. Another thing, like squirrels, like there's red squirrels and there's brown or gray squirrels, and how many of those you see in a certain area over time. If the gene frequencies don't change, then microevolution has not occurred. So there's this thing called Hardy-Weinberg equilibrium. These guys, Hardy-Weinberg, came up with the idea. We'll talk a little bit more about Hardy-Weinberg in a moment. But Hardy-Weinberg equilibrium is this idea that if these certain conditions are met, then you're not going to experience any change in the population. But this is just kind of the settings that are required in order for Hardy-Weinberg to be met. So Hardy-Weinberg can sometimes be abbreviated HWE. Allele frequencies in a population will remain constant, not change, if there's no mutation, no migration, a large gene pool, random mating, and no selection. So all of these conditions have to be met in order for equilibrium to be achieved. Now, the thing with this is it is very, very, very rare for this to be achieved. In fact, it's almost impossible. Sometimes you can achieve this in a laboratory setting, but even then it's very hard because you can very rarely account for no mutations. In a lab, you can usually get no migration to happen. You can sometimes account for random mating and no selection. You can sometimes do a large gene pool in a lab, but it's very rare to make no mutations in a lab. So even in a lab, it's very hard to get Hardy-Weinberg equilibrium to occur. So this is a very, very rare thing to happen. And in nature, in a natural environment, it's almost impossible for Hardy-Weinberg equilibrium to occur. But in order for there to be absolutely no changes in a population, all of these conditions must be met. There must be no mutations happening. There must be no migration, no populations moving into or out of the environment. Has to have a large gene pool to pull from. Has to have random mating taking place. And it has to have no selection, like no pickiness. The animals can't be picky about who they choose. And no, like, natural selection taking place. So in this case, we have 1 to 2 to 1 ratio. And in order for this ratio to remain the same, in order for this to stay the way it's supposed to be, then we have to meet those conditions. Actually, now that I look at this, it's not a 1 to 2 to 1 ratio. Because it's 0.04 to 0.16 to 0.64. That's not a 1 to 2 to 1 ratio. But in order for this condition to be met, Hardy-Weinberg equilibrium would have to be consistent. So required conditions are rarely, if ever, met. Deviations from a Hardy-Weinberg equilibrium indicate that evolution has taken place on a micro scale. So anytime we see that the allele frequency has changed, that means that some kind of microevolution has taken place. An example of the birds eating the moths is a really good example. Some moths are going to camouflage better than others, and the birds are naturally going to eat the ones that camouflage less. And so the ones that stand out are going to be the ones that the birds eat. And then we're going to see a shift in the color of the moths that live to reproduce. So if the moths are going to eat the light-colored one, like this bird, or the moths, the birds are going to eat the light-colored moths. This bird is eating the light-colored moths, and so the dark ones are going to survive. They're going to pass on their dark-colored alleles, and so the population is going to shift to the dark-colored moths, and so we see a shift, and that's considered microevolution. Now it is microevolution because they are the same moth. We just have a shift to a different color formation or color trait, and they're the same moth still. They can interbreed, they can make more offspring. It's just now we have a trait that's standing out more than the other. So over time, we're going to see a population change in the frequency. The mating will be and here's each of the Hardy-Weinberg conditions. So was random mating. Alleles do not sort randomly, so evolution did not occur here, but evolution did occur in these cases because there was selection, there was mutation, there was immigration coming in, immigration leaving. So two types of immigration. One of them is leaving the population, one of them is coming into the population. And then there was other types of bottleneck and founder effect. So genes being changed depending on availability of the genetic information in the population. So looking at mutations available, genetic mutations are the raw material for evolutionary change. They provide new alleles. Now remember, thinking back to mitosis and meiosis, we have the ability to cross over during prophase 1 of meiosis. We also have mutations that can take place in many parts of mitosis, such as insertions and deletions, and those kinds of things. They can provide new alleles. Some mutations might be more adaptive than others. Affecting pigment color in peppered moss have provided the variation needed for natural selection to occur. So some mutations can be beneficial, some can be detrimental, some of them can be neutral and not cause anything, but mutations affect the actual genetics of the organism. Gene flow or gene migration, the movement of alleles between populations, such as seeds that are carried into another population. Breeding individuals migrate into or out of a population. Movement of genes allow resistance to herbicide. Continual gene flow, reducing divergence. The migration between populations doesn't occur. The gene pools become more different over time. And differences in genetic makeup of these populations become so large that they become reproductively isolated. So if you have a flood that divides a population in two, or an earthquake that creates a like a ravine or something between two populations and separates them, then there's a lot of things that can divide a population into two, and that separates them so that now they are isolated from each other, and they will eventually develop into two completely different populations, and those two will become reproductively isolated and start breeding amongst themselves, and that could lead to two individual populations. So here's an example. There's a river here, and some of these seeds could get blown across that river or carried across that river by a bird. Like the seed gets eaten, the bird flies across and defecates the seed out onto this side, and it plants over here and grows over here. And so the population that grows over here is different than the population that grows over here. So this is an example of gene flow across the river. So genetic drift is changes in the allele frequency by a population due to change rather than selection by the environment. Does not necessarily lead to adaptations to the environment. So it doesn't always mean that the environment had an effect on it. It could be due to just other things. Caused by disproportionate random sampling from the population can cause the gene pools of two isolated populations to become dissimilar. Some alleles are lost and others become fixed or unopposed. So this is something that can happen after a bottleneck when severe inbreeding occurs. A good example again comes back to the black-footed ferret. So black-footed ferret is an excellent example of inbreeding and the bottleneck effect. The black-footed ferret was thought to be extinct, and in Wyoming, a farmer's dog brought to it a animal in its mouth, and the farmer was wise enough to see that this was an animal that it didn't recognize, and contacted the local authorities and discovered that it was a black-footed ferret, and that there were 7 or 8 that they discovered on its farm, a last colony. And black-footed ferrets live with prairie dogs, and so they live in the prairie dog burrows, and they survive and eat the prairie dogs. And so the conservation people collected the black-footed ferrets and bred them in the lab, and took extreme conservation methods. And because of that, they went into this captive breeding program and re-released many of them back into the wild on prairie dog sites, because they have to live with the prairie dogs in their burrows, and their sole food source are the prairie dogs. Now there are hundreds that have been re-released, and they are doing much better. But because they came from that original 7 or 8, there's only 7 or 8 genetics to draw from, and so they have a severe bottleneck of their genes, and they have a severe inbreeding problem. And so there are a lot of mutations and genetic diseases and disabilities that the black-footed ferrets have, because all of the genetics are coming from those original 7 to 8, and there's not really much that we can do about that until the population recovers, because they're all drawing from the same genetic pool. So there's a stronger effect in smaller populations because they need to build up over time, and over time it'll get better because they will have mutations that compensate for the original bottleneck effect. But until they're able to build up those genetics again, their gene pool is quite restricted. So here's an example of population drift. 10% of the population, the forest fire kills 5 light moths, and then 20% of the population is going to kind of replenish that area. And so we have these moths that are coming back, and we have a darker moth that is found there. But it is, there are less now. There's less of the lighter moths, and the darker moth is more prevalent, and because there's not as many of those light moths to kind of override the dark one. So the bottleneck effect again, a random event prevents a majority of individuals from entering the next generation, such as the deaths of many of the black-footed ferrets. Many of the prairie dogs were killed off because farmers didn't like the prairie dogs, and so because of that, the black-footed ferrets went with the prairie dogs, and so many of the black-footed ferrets were destroyed. The next generation is composed of alleles that just happened to make it, and is mostly due to natural disasters or habitat loss. In the case of the ferrets, the prairie dogs were killed, so the ferrets again went with them. Now there's also the idea of the founder effect. A new population is started from just a few individuals, so a few individuals kind of went off and started their own kind of group. The alleles carried by the population founders are dictated by chance. Those few that started the new group are the ones that have the alleles that started this new group, and so the gene pool of the small population is much different from the gene pool of the original population. The greater the reduction in population size, the greater the effect on allele frequencies. So the alleles that used to be rare will start to occur at higher frequencies, or it might be absent altogether because maybe the founders didn't have those rare alleles at all. So if they didn't have them at all, they're not going to show up at all. So this can lead to a higher than normal occurrence of inbreeding, and because if the founders were siblings or cousins, then there's no choice but for them to mate with each other. This leads to a higher chance of rare recessive disorders emerging more frequently because they just don't really have a choice. If they want to continue to survive, they're going to have to breed with their siblings. So here's an example of bottleneck and founder effect. 1 marble equals 10 alleles. If you only have so many genes to pull from and they get stuck coming out of the bottle, and you try and it's restricted, only certain alleles can come out of the bottle, you don't have as many that you can pull from. So only a few of the alleles can come out, and the offspring doesn't have a choice. Some of those rare alleles might not be able to get out and be expressed, and only the more common ones are really going to be expressed. Non-random mating is another issue when individuals do not choose mates randomly. Sometimes the female only wants to mate with certain males, and so the males that are not as enticing are the ones that are not chosen, and so they don't get to pass their genes on. And so if a female is being picky about the males that they choose, only certain males get to pass their genes on. So with assortative mating, individuals select mates with the same phenotype with respect to a certain characteristic, like the peacock with the very fancy tail. And so only the male peacocks with the really fancy tails get to pass their genes on. So individuals reject mate with different phenotypes, and so this is restricting the genes again to select a male with a very fancy tail. And so any other characteristic is lost over time in the population. So it can play an important role in the evolution of a population. So on the island of Pingelap, non-random mating resulted in an increased frequency of color blindness because that particular trait, whatever trait they were looking for, those males had color blindness as well, and so they kept selecting for those males, and the color blindness trait kept getting passed down as well. So moving on, talking about natural selection. Natural selection is the adaptation of a population to the biotic or living and abiotic non-living environment. There are variations, then members of a population differ from one another. Inheritance, differences are heritable, genetic differences. Excuse me. And differential adaptiveness. Some differences affect survivability and differential reproduction. Some differences affect the likelihood of success in reproduction. Natural selection favors the variant that is most adaptive to the present environment. Means survival of the fittest. That's basically what it means and what it sums up. Those with the best traits are going to be the ones that are able to survive in their particular habitat. If you take a penguin and put it in the desert, it is not adapted to survive in the desert, so it is not going to do best. So it's important to remember that animals that are adapted to survive in their habitat survive in their habitat best. It's not going to survive in some other habitat. They are able to survive in their habitat the best. So there are three types of natural selection. You've got, this is a range of phenotypes. Remember, phenotype are physical traits, genotype are genetic traits, and you've got different kind of directional shifts. So when natural selection happens, you can select for the middle trait, the trait that occurs here in the center. You can select for an outlier that shifts this direction, or you can select for the outliers that are on either end, like we see here. So in this case, the trait that is in the middle is selected for, and so the graph will shift so that we're just selecting for the middle trait. Or we can select for a trait on either end. It can go to this end or this end, so the entire graph shifts to this side, or the entire graph shifts to this side. Or we have disruptive, this is called disruptive, where we have shifting to both sides and the middle kind of disappears. So we have directional, shifting to this side or this side, and we have stabilizing, where it shifts in the middle. So three types of natural selection: stabilizing, directional, and then disruptive. Most traits are polygenetic, poly meaning many, variations and traits that result in that bell-shaped curve. So this is a bell curved. It looks like an upside down bell. Well, I guess it's not upside down. It looks like a bell-shaped curve. So most traits are polygenetic variations and result in a bell curve. And then again, three types. So stabilizing was this first one we talked about, where it's going to shift to the middle. And this means that the middle trait, whatever is the most common, In the middle, that's going to be what is the most adaptive to the environment. Human babies with low or high birth weights are less likely to survive. So it's that middle kind of average birth weight is the one that is most likely to survive. If a baby is too big, it usually has health problems or trouble with birth. If a baby is too little, it has health problems and not able to survive. So it's those average birth weight babies that typically do the best. So that's a good example of stabilizing selection. So here we can see how that works. So this is birth weight, and this is percent of births in population, and this is percent of mortality. So babies that are in the middle are the ones that are going to survive the best. So the ones that are in the middle are the best at surviving. So this is stabilizing selection. Then directional selection, again, is this shifting all the way to one side or shifting to the other. So that is stabilizing selection, shifting to one side or the other. An extreme phenotype is favored, and the curve shifts either to one or the other. Bacteria become resistant to an antibody is a good example of directional selection. And then disruptive selection is like picking one on either side, and then the middle is kind of cut out. So two or more extreme phenotypes are favored, and the middle is not selected. So British land snails vary because a wide geographic range causes selection to vary. So here we have directional selection. So we have resistance to antibiotics. After time, it has caused it to shift, and it's caused a shift to drug-resistant mutants, and so it's shifted to this kind of yellow one over time. And then here we see disruptive, where we have a banding pattern, and it has caused two kind of distinct, and this middle has disappeared. So we've gone from this extreme to this extreme, and the middle kind of blended one is gone. So this one's disruptive. It's disrupted the graph. All right, so sexual selection. This is another, going back to that Hardy-Weinberg again. Adaptive changes in males and females lead to an increased ability to secure a mate. With males, there is an increased ability to compete with other males for a mate, so they are more able to compete for a mate. And females choose to select a male with the best fitness, ability to produce surviving offspring. So that's kind of what the girl wants. She wants proof that the male has healthy genes and is going to produce healthy offspring. The female has to put in a lot of work to take care of the young because the male doesn't always help, and the female is going to have to do most of the work most of the time, and so she wants proof that the genes that she is left with is going to be able to give her healthy babies. And so being able to survive is called fitness, and so she wants to see that the male is going to provide her children and her offspring with a good fitness. So the choice of a mate is a serious consideration because females produce few eggs. So she's only got a couple times that she can choose to get a good mate. So there's this good genes hypothesis. Females choose mates on the basis of traits that improve the chance of survival. There's the runaway hypothesis that females choose mates on the basis of traits that improve male appearance, that if a male looks good and healthy, then that means their genes are going to be good and healthy. Sexual dimorphism, males and females differ in size and other traits. And females' choice can explain why male birds are often more ornate than females, because if a male has fancy colors, that means that he doesn't need to camouflage. He can afford to stand out and still find food and still survive. Remarkable plumage of males may signify health and vigor, that if they look fancy, they don't have to camouflage, they are strong enough and healthy enough to stand out and find food, and they can beat predators while doing all of that. Birds of paradise are one of the best examples of these. This is a male, this is a female. The females often have more camouflage because they end up sitting on the nest. The males look all fancy because they can avoid predators and they have more energy to dedicate to their fancy colors and to their fancy plumes, so that way they can attract a mate, and only the healthy, well-fed males can afford the energy necessary to devote to that. Males can father many offspring because they continuously produce sperm in great quantities. They compete to inseminate as many females as possible, and so they will fight for the attentions of the female, and the females may end up mating with more than one male. Sometimes they can be inseminated by different males, and they can carry offspring from more than one male. Cost-benefit analysis is access to mating worth the competition. So this is a male displaying full threat to other male baboons to try and protect his territory and to guard his females. Here's some competition. Male deer will grow antlers to compete with other males and also to protect their females. So populations with limited variations may not be able to adapt to new conditions. Adaptation is vital for any species or any population to survive over time. They need to be able to have a good variety of genes. Now, it's not always possible for an individual to survive, but the idea is that if there's enough diversity, the population as a whole can survive. So maintenance of variability is an advantage to the population. Again, an individual may not survive, but if there's enough diversity, then the population as a whole may survive. So I may not live, but my neighbor might. And as a species, that's good. Natural selection causes imperfect adaptations. It depends on evolutionary history. Evolutionary does not start from scratch. It takes what's already there and builds off it and uses the diversity that's available to hopefully change and modify things as time goes on. The process of development restricts the emergence of novel features. Imperfections are common and normal. Things aren't always going to go the way they're supposed to. There's going to be mistakes in evolution, and that's part of the process. The benefits of freeing our hands outweighs the cost of spinal injuries from assuming an erect posture. Many animals are quadrupeds. They walk on 4 legs, and that makes it easier on their spine, but it makes them use their hands. Us being upright does lead to more spinal injuries, but it does free our hands up to do many more things. By having our hands free, we're able to manipulate objects and carry things and move things around and do many more things than organisms that need to have their hands. So that was a benefit to us to become bipedal and use our hands and free up our hands to do other things with. So disruptive selection due to environmental differences promotes polymorphism in a population. Population occupies a wide range and may have several subpopulations designated as subspecies. So this is where it gets a little gray area. Some scientists don't like subspecies. Some argue that you should just have a species and not divide it down into subspecies. It really just depends on the scientist. Environment includes selecting agents that help maintain diversity. Again, diversity is good. The more different kinds of things, the healthier the population is. Maintenance of variation among a population's members has survival value for the species. And an example would be an average beak size of Galapagos finches depends on the available food supply. Lots of different kinds of available resources and lots of different kinds of available things makes for a better chance of survival overall. Here are just some examples. Subspecies help maintain diversity. Different kinds of this, these are all the same species, these are just different subspecies. And so a variety of the same kind of snake is going to help the snake survive under different conditions. And going back again, diversity is good. So recessive alleles often hide in a population, and that's because the heterozygous individual hides the recessive allele, and it prevents it from being fully expressed. The recessive allele is still in the population, but the dominant allele will hide it when you have a heterozygous individual. A heterozygous allows even lethal alleles to remain in the population. They're still there, you just don't see them, and they can remain there unless two heterozygous individuals come together, and even then you have to have, if you do a Punnett square, only 25% chance that the two recessive alleles will come together, and there's 75% chance that the individual would not receive the two recessive alleles. So it's very rare that the recessive alleles would actually come together like that. So the heterozygous advantage, it assists the maintenance of genetic and therefore phenotype variations in future generations. Sometimes recessive alleles confer an advantage to heterozygous. Sickle cell disease, heterozygous individuals don't die from sickle cell disease, and they often don't die from malaria. Sickle cell anemia alleles is detrimental in homozygous. However, heterozygous are more likely to survive malaria. So if you have sickle cell, but you have it as a heterozygous form, and it's hiding on that recessive allele, your dominant allele overrides it, you are more likely to be okay if you got malaria. So that's kind of an advantage of that. The sickle cell allele occurs at a higher frequency in malaria-prone areas. So if you have malaria on you're more likely to have sickle cell if you're heterozygous. So it's kind of a it's an advantage to have that. So it's kind of interesting how our bodies work and how our bodies figured that out. This is a form of stabilizing selection. So it's kind of a negative thing if you have the homozygous recessive, but it is an advantage if you have it in the heterozygous form. So malaria, sickle cell, and then an overlap of both. All right, well, that ends this chapter, and we will see you to the next one. We are getting close to the end and moving right along. See you next time, guys.